So yeah, the presentation today I was going to go through is a high-level overview of 3D cell cultures, talking about some of the applications that our lab has worked on, and also diving into tissue clearing and how it applies to 3D cell cultures. It's something we've heard about in whole mouse brains, whole rodent brains, but how it's actually important to looking at 3D cell culture models will be a focus of this talk. And I'm going to dive through some real-world examples of how we've applied tissue clearing and how we've looked at 3D cell culture models. Now, just some background on us. Our company is a contract research organization that's spun out of Rutgers University, and we are focused in the area of 3D imaging, digital pathology, and automation with a strong focus on 3D cell culture models. Basically, the focus of our company is turning tissues into insights through generating lots of data through advanced imaging. Now, for this presentation, I'm going to give a background on 3D cell culture. I know it's been a topic of interest today, but I'm going to take it at a really high level, and then dive into background on high content screening, some limitations of high content screening and the imaging of 3D cell culture models, and lastly, dive into some case studies. Now, this is a question that's come up a bunch today, and a lot of folks have gone over why 3D cell culture models are important and why we're using them, but just to kind of back up here, traditionally there's a relatively poor translational gap between in vitro results and in vivo results. And the whole idea behind 3D cell culture models is to improve this translational gap. And this translational gap has some real-world problems resulting from it, first of which is reduced in vitro selectivity. Expanded in vivo studies, so if we have poor selectivity in vitro studies, we're pulling in many more compounds than we might want to for expensive animal studies. We have later stage failures and unnecessary screening, and lastly, increased development costs and time associated with in vitro studies. Now, just diving into this a little bit more, it's obvious, this slide I show for anybody who's in the space of in vitro assays is just looking at the human body. The human body occurs in 3 dimensions. We have gradients, we have diffusion, we have all different features that happen in 3D that do not happen when cells are just placed on a plate in 2 dimensions. So 2D cell culture models, they have many applications, which I'll talk about, but 3D cell culture models are coming into a whole bunch of different fields. In the last 5 or 6 years, they've gone from a very basic research application, a few publications really focused in academia, to at the forefront of drug discovery and being adopted by numerous pharmaceutical companies. Now, this is something we kind of bounce around with the nomenclature. You hear these terms all over the place out there in papers. There's not really a consensus around these terms, so I'm just going to talk through them. Spheroids, what we generally refer to as spheroids, are a broad term for 3 agglomerations of cells, a few cells that are together in a spherical shape or so. Microtissue is a term that was coined by Sphero. The idea here is that they replicate the in vivo characteristics of tissues. And then lastly, we hear organoids a lot. And these terms are kind of used interchangeably, but they all fall into the bucket of 3D cell culture models. And that's a term that we use. For me personally, I think organoids is taking a step too far. We're not quite at miniature organs in a plate. We're not really even close yet. So I think using that terminology is a little misleading, especially as the folks not working in this field. So we always use, and I'll use today, 3D cell culture models. Now, for generating 3D cell cultures, there's a whole bunch of different techniques that are out there. We have Matrigel, we have ultra-low attachment plates, we have the magnetic printing system from N3D, which works quite well, and then the hang and drop system, which Sphero has focused on a lot. And what's important to understand, and what I'll talk about a lot today, is that each one of these techniques has its own specific use cases. We have advantages and disadvantages based upon what we're trying to do. And I think it's really important to call this out, and also to call out that all models are not the same. We don't always want really, really complex models, depending upon the type of assay that we're trying to develop. So based upon a research question that a group is going after, for us it really depends upon the model, the project, the throughput, the cost will vary based upon the type of assay that we're actually going towards on what system we're using to generate 3D cell culture models. Now, this is another really important slide, and this drives the point home that there are advantages and disadvantages, and 3D cell culture models are not a replacement for 2D cell culture models. I think there's a lot of companies that are trying to sell 3D cell culture models on a plate and say, get rid of all the 2D stuff you've done before, moving to 3D, but I think that is sort of an improper justification, because 2D is a balance with 3D as far as cost and throughput. 2D is always going to have higher throughput. It's always going to have lower cost than 3D. So it really depends upon the application and the type of question you're trying to answer. But it's always a balance between these three parameters, so cost, throughput, and relevancy. As we approach animal models, we get into clinical testing, the relevancy goes up, of course, but along the way, the cost and the throughput are going down. So it's really a balancing act, and where 3D cell culture models fit in is between 2D and getting into animal models and trying to improve those assays and their accuracy before we start spending more and more money on expensive animal studies. So it's really a balancing act, and it really depends upon the assay that you're trying to develop. Now these are just a list here of a lot of points that came up this morning, but advantages and disadvantages of 3D. So the strengths are the relevance. We're improving the physiological relevance of the model compared to a traditional 2D model. We have the morphology, the cell phenotype. We're getting better predictive power of what's going to happen in vivo in regards to efficacy and toxicity. We have heterogeneous cell populations that better mimic what's going on in the microenvironment within the body. And there's lots of reagents that are out there today for growing 3D cell culture models. Over the last few years, it's become easier and easier to get these models, to grow them, to validate them, and to use them in the research, just as the technology started to explode and really come into the spotlight. But the weaknesses are that because it's new, it's a nascent technology, there's not consensus in the marketplace. There's groups all over the place doing all different types of things, using different nomenclature, different processes, different procedures. There's a liver model over here, there's a liver model over there, and there's not consensus in the marketplace yet around what model is best for a certain application. So we see a lot of variance out there, a lot of groups doing a lot of different things. So the space is relatively new. And a lot of companies, especially Thermo, stem cell technologies, a lot of companies that are out there are trying to build the consensus around what models are best for certain applications, but the space is still quite new and still quite immature. Now to go over the traditional methods and why I think this is really important is just to show again that it's about the model, it's about the system we're using, but it's also about the type of data that we're trying to get from one of these 3D cell culture models. A lot of folks out there will go today and do a dissolution assay, so something like total ATP from one of these models. The problem with that is that it's not going to generate a whole lot of data. All the three-dimensional data that's embodied within that model, you're losing by getting that single endpoint from that assay. But if that's the type of question you're trying to answer with your research, it's totally fine, but it's really a balancing act here between cost and throughput and the amount of data that you're getting from one of your assays. From a 96-well plate of spheroids, you can get terabytes of data. You can get a few endpoints. You can get a one-page report, or you get hard drives of data. The amount of data you're collecting really depends upon, again, that research question that you're trying to go after. So we're stretching here from just a dissolution-based assay, a couple endpoints, all the way up to the three-dimensional imaging of these models and generating huge amounts of data. And in between, we have traditional histopathology, so sectioning 3D cell culture models, which is very tedious, very time-consuming, and also very expensive. But at the end of the day, it all comes down to the research question you're trying to address. Now, one of the limitations with imaging of 3D cell culture models is that they are in 3D. And the problem with that, as you see up here on the slide, is if I'm looking at a model, this one here, which is not transparent, it's opaque, it's just sitting in PBS on a well plate. If I try and image it with confocal microscopy, and I move up, let's say 10 microns through it, taking different Z sections as I'm moving, the interior of that 3D cell culture model is dark. And we as a company, when we first got into this space, saw an image like this at a conference, and we asked the researcher, why is the center of your model dark? And they said there's nothing there. The thought for a long time was that these 3D cell culture models had a totally necrotic core, nothing was there, they were totally hollow. But what's actually happening is light is not penetrating into these models because past 1 or 2 or 3 cell layers, light is attenuating, and it looks like we have this eclipse. But if we take this exact same model here, which is just labeled with the nuclear stain, and we go ahead and clear it, we go ahead and actually acquire pretty much all the data throughout the depth of that model. So we're filling in the interior. The reason this is important is you're running some kind of drug screen or any assay really, and you're only characterizing the exterior of these models, you're biasing your results towards the cells on the exterior, which are vastly different than the cells on the interior of the model, which I'll show you in a little bit. But what we did here is we took these 3D cell culture models and we cleared them with CytoVista 3D cell culture tissue clearing reagent, which allowed us to increase the amount of cells we're detecting by about 3-fold on Widefield and about 3 to 4-fold on Confocal, which allows us to actually characterize all the cells in the model, again, instead of just the periphery. And to show you graphically what this looks like, as we're going from the bottom of the model up, you can see this difference is pretty significant. So in basically the center of this 250 micron diameter model, we're almost characterizing about 6 or 7 times more cells when we clear the model than we do when it's not cleared here. Now, as a contract research organization, we find it really, really important to stress the details with all of our clients. I think this is important for anyone developing an assay in this space, is to stress the details on what kind of data we're going after and the type of model. These are the questions we always ask clients that we're working with. I think are always really important to run through. But as a type of model that you're going after, we have highly complex 3D models that are out there. They're thousands of dollars per well plate. We have models that we can generate for $50 to $100 per well plate and everything in between. And really it comes down to what is the throughput of the assay we're going after and what is the type of data we need to get. We can spend a lot of money but not answer a question better than we spent a little bit of money on the model that we're using for the assay. So that's an important consideration up front. Are we just growing Hep G2 cells or are we growing multiple types of liver cells in a model? Next part is identifying the minimum amount of data that we're trying to generate. This is really important if you're developing an image-based assay. If we're doing 4x versus 40x, and we do 1 micron versus 10 micron z-steps, the difference in data can be 100,000-fold based upon the parameters that you're extracting from the tissue and how much data you're acquiring. So these questions are really important in defining what type of data you're acquiring and how long the assay is actually going to take you. There's this, I think, misbelief that more data is always better, and in this space it's definitely not true. It's about getting the data that's able to answer your question and not much more than that. The next part is labeling, so choosing the right labels to actually go ahead and acquire the targets. Relatively straightforward. Optimization has to come into play to make sure you're getting uniform labeling throughout the models. And then lastly, the data distillation. So you can generate terabytes of data, but you have to turn it into some kind of actionable insight at the end of the day. And we find it very important to know what that insight is before we even start, so we can define it down to that specific table or that specific endpoint that a client's looking for. Now, for labeling with tissue clearing, there's a whole bunch of different tissue clearing techniques that are out there. The CytoVista tissue clearing reagent is designed for 3D cell culture models. It's designed for high throughput and automation, and it's compatible with fluorescent probes, immunolabeling fish, and fluorescent protein. There was another misconception in labeling 3D cell culture models that labels couldn't penetrate to the center of these models. Now, our lab started out with actually clearing and labeling whole mouse brains, very large tissues, getting antibodies across 3, 4, 5 millimeters in depth. So the issue is not actually getting antibodies across these tissues. It's about optimizing and getting it there and coming up with a process that works for your tissue. So we always suggest an optimization process where we go through and we titrate and we look at incubation times for each individual model, and we optimize that based upon the labels that we're trying to go after. But all these techniques are possible, and achieving uniform penetration and labeling of models is possible with antibody labeling, and of course the small molecules up here that we're talking about as well. Now, another consideration is the way we're actually going ahead and illuminating the model. We have the opportunity to look at the CX7 LED and the CX7 laser in our lab. And for us, we wanted an imager that wasn't only used for 3D cell culture models, actually able to be used for whole mouse brains, about 1 millimeter sections, human biopsies, a whole bunch of tissues outside of just 3D cell culture models. And to image deep into whole tissues, we knew we needed a system that had laser-based excitation, because if you have an LED-based excitation source, you're going to get a lot of diffraction throughout the depth of the tissue, and the imaging artifacts are going to be very significant past about 50 microns or so of depth. So what I'm showing over here is just the CX7 LED. There's a little bit of an artifact here, which was on one of my previous slides from the LED. This is about 130 microns, 120 microns into the tissue. It's a little bit of shadowing, but with the laser-based system, you don't get that. It's a much crisper image throughout the depth of the tissue, and we've actually used the CX7 LZR system to image about 1.5 mil through tissues with just standard air objectives. So the ability to get depth with laser-based excitation is just much better. We have a number of systems in the marketplace today. There's the Opera Phoenix, there's the GE InCells 6000, 6500, the CX7, and then for the LED, we have a number of devices that are out there, but for us, we always, when we're going out and looking for a device, we're always trying to find a device that had laser and not LED. And there's this other interesting thing I wanted to mention, which is this foreshadowing. A lot of folks use the Corning U-bottom plates, which make 3D cell culture models really, really easy to grow. The problem is if you're using an LED-based system, you get a lot of what we call foreshadowing below the model. So there's like a weird kind of light shadow that bounces off the bottom of the plate and casts itself onto the bottom of the models. So you get this like halo effect on the LED systems, which you don't get in the laser system. So if you're using that system, that's another consideration to just keep in mind. The other consideration we've heard a lot about from researchers is the type of objective to use on the microscope. There's a number of immersion objectives that are out there. Immersion objectives are very popular if you're trying to image into a full mouse brain depth, because you absolutely need them to get past 1 or 2 or 3 millimeters of depth into a tissue. Otherwise, you get a whole bunch of problems with refractive index mismatch. But for looking at 3D cell culture models, you don't need a dipping objective. There's the PerkinElmer Opera Phoenix instrument has a dipping objective that's actually inverted, and it works well. You get definitely better images by doing that. But if you clear a tissue, the difference between an air objective and a dipping objective isn't as significant as it is with just an air objective versus a dipping with no tissue clearing. So it's kind of a myth. You don't need those expensive objectives to actually improve your image quality. You can just render them transparent and very easily just get a much, much crisper image by better matching the refractive index throughout that model. And this is true to about 1 millimeter or so in depth. It depends on the model, but about to 1 millimeter or so with air objectives, you can get a very crisp picture on most inverteds, whether it's the CX7 or whether it's one from Zeiss or Leica or whomever. The other consideration is confocal versus Widefield. As we're developing assays, this is a really important consideration because this dictates how much data we're generating. So are we generating hundreds of pictures throughout the depth of a model, or are we getting one single image from the whole model? The reason this is important is because many assays don't actually require three-dimensional information from these models. Some do. But this really will define how much data you're generating and the throughput of your assay. Having to do confocal imaging on an entire 96 or 384 well plate is going to take way, way longer than doing wide fields. This is one of those questions that we always try and knock out up front, is that data density that we're trying to get from our models. The negative, of course, of wide field being that we're not getting depth information, but depending on your research question, it might not actually matter. Now, another thing we see pop up a lot is this confusion around Z projections. Most of the data that you've seen around 3D cell culture models in publications is typically a Z projection. So all the Z stacks taken from a model are just compressed down to one image. And that's part of the reason why a lot of folks never noticed this ring effect, is they never looked, they might have looked, but might have ignored it, that you look at the 3D cell culture model in Z slices, you see the rings. But if you compress all those down into one Z projection, you don't see that. So a lot of researchers process their data from Z projections as one single image, and you miss that bias by doing that, whereas most of the work that we do in confocal imaging, we actually process each individual Z slice individually and not actually project those down to one single image. So this is an important distinction, but again, depends upon what you're trying to do. There's plenty of assays that we do where we do use Z projections, but if we're trying to do something like measure antibody penetration into a model or look at immune cell invasion, it's something we want to go ahead and look at Z stacks for. Now, the first case study I was going to dive into is really digging into why 3D is important. A lot of folks just say 3D is better, and it's good to understand that there are some specific case studies on why just growing models in 3D is actually better. So what I have on the screen behind me here is a wood cell line. It's from a company called Solaria. It's up in Boston. This wood cell line expresses ER from the patient it was derived from. And what we've shown here is by growing a model in 3D versus 2D, we actually get almost no expression of ER in 2D. But when we grow this exact same model in a 3D cell culture, this was in a Corning U-bottom plate about 400 microns or so in diameter, we actually do get very significant ER expression. So the expression of biomarkers changes in many cases dramatically between just growing in 3D versus growing in 2D. And this, the thought is, of course, that we're more accurately replicating what's going on within the body. And because of that, we have a more accurate representation of protein expression compared to what's going on in a 2D cell culture model. Now, this comes into play here as we look at a whole bunch of different compounds and we start exposing those 3D and 2D models. So over here we have a 2D in blue, and we have the 3D models over here in red. So if we take fulvestrant and we go ahead and take this ER targeting drug and we expose 2D and 3D, both this exact same drug, We don't see any real response in the 2D models, but we do actually see a response in the 3D models, as to be expected, because we're expressing ER in one but not the other. So the impact here is that we're getting dramatically different results screening the exact same drugs between 2D and 3D, just based upon the physiological properties of what's happening in 3D versus 2D. So this is one just very simple example, but the point here is that 3D does improve many assays depending upon the type of research question that you're going after. And then again, looking at another example here is just tamoxifen. So we go again and see that based upon growing in 2D versus it growing in 3D, just a small screen here, that we do a very differential impact based upon what this drug is actually indicated for. So we're looking at ER expression again, and the change here is again due to that expression between 2D and 3D. So the second case that I want to dive into is what is at the center of a 3D cell culture model. This is sort of a myth that I would say we dispelled with some other researchers, is that not all 3D cell culture models are hollow, not all of them have a necrotic core. That was always a thought that after maybe 50 microns, we're starting to get blackness in the center of that model, that it's necrotic. It's not necessarily true. What we did is we grew up Hep G2 spheroids, we grew them in ultra low attachment plates. Went ahead and used the Thermo Live Dead Kit with DAPI labeling. We used the CytoVista tissue clearing reagent to go ahead and actually clear these Hep G2 models, and then we imaged them in 3D using the CX7 LZR. And what we showed down here is that based upon the seeding density of where we started with, we get significantly more necrosis, of course, with seeding with more cells as the models are getting larger. And these do have a necrotic core, but it's not a hollow necrotic core. It's not that there's nothing there. There are cells there. Those cells are just dead in this case here. So what we've shown in a whole bunch of different cases is that there's always typically something at the heart of these models. Sometimes we get very large. Those cells do start to die and regress, and there might be a hollow core to some of the models, depending on how large they are and how old they are. But there is, in fact, something at the center of 3D cell culture models. This is the question we get a lot, is that why would I ever want to get 3D data from a model? Yes, 3D cell culture models more accurately represent what's going on, but I just want to get the data, just the same endpoints I've always gotten. Why is 3D necessarily better than 2D? Again, we took Hep G2 cells in this case study. We seeded at 1,000 cells a well, again, ultra low attachment plates, which are just really, really easy to use. We dosed with Taxol at varying concentrations. On day 4, the spheroids were fixed and processed. And we took the same spheroids, we cleared them again with the CytoVista tissue clearing reagent. We labeled with Ki-67 in this case, Alexaflor 488. And we went ahead and looked at cleared versus non-cleared. And what we're showing here is there is a significant difference between the two. I'm going to start on the right here, of looking at Ki-67 positive cells. On the cleared, our slope, our percentage here is much lower. On the non-cleared, it's much higher. The reason for that, and the non-cleared models that we have, we're only really looking at the periphery of those models. We expect that percentage of Ki-67 cells to be the greatest anyway, but when we're actually characterizing the entire population and not just that small percentage on the outside, we see that number come down quite significantly. And if we look at the with respect to the vehicle over here, that difference is very significant. And we get very much higher sensitivities based upon clearing versus not clearing, as we're increasing that population of cells by about 3 to 4-fold, and we're actually characterizing that entire population. So getting at, again, why this is important and why 3D data. Now, what we can do is, because this data you can pull into any kind of n-dimensional matrices that you want, you can actually start bidding the data at different parts of these models. So we can look at the inside and the necrotic core, perhaps, the middle of the model, the outside of the model, and we can show, based upon where we are in these 3D cell culture models, what's happening. Now over here, we're just showing Ki-67 as a function of where we are in the model, but the reason this is important, a lot of the work that we've done is around things like antibody penetration. So for very large molecules, we think they have great efficacy in a 2D cell culture model because they're hitting the 2D cell culture model, but we put them in a 3D cell culture model, and they're getting to about here. So we think they have great efficacy in 2D, but they're not really touching the middle or even the center of those models. So when we look at different antibodies, different fragments, different types of antibodies, we can actually quantitatively show that some are actually better at penetrating into the models. So if we have a solid tumor model, we want to make sure that our antibody is actually getting to where it's supposed to go and understand the kinetics of that penetration. So this is why, and it's not always the case, but there are cases where generating 3-dimensional data and understanding in a 3-dimensional matrix what's going on is incredibly important. So a lot of this data that we process and we generate, we actually do outside of the CX7 software, just in regular ImageJ and cell profiler pipelines, but it allows us to take the data and really play with it however we want. It comes out as TIFF files, and we process it, turn it into these insights for our clients. Now, if you want to reach out to me, my info is up here, and I look forward to your questions. Thank you.